In an earlier video on genetics, we talked about how traits were passed on across generations through particles of inheritance. Today, we know these particles as genes, which are made up of DNA and responsible for carrying genetic information. In this video, I want to explore what these genes are, what they do, and what they mean for life on this planet. Picking up from the last video, the rediscovery of Mendel by Thomas Hunt Morgan, as well as other biologists, sent shockwaves across the field. Everyone was searching for the true nature of these mysterious particles, and Morgan, for one, was going in the right direction. One of the theories of the time pointed towards squiggly little cellular worms that danced about and arranged themselves in a way reminiscent of Mendel's particles of inheritance. These were called chromosomes, and according to this theory, they housed the particles of inheritance and allowed them to be passed on to offspring. However, this clashed with Mendel's law of independent assortment, which dictates that the inheritance of one particle has no bearing on the inheritance of another. Consider a plant with both types of particles for pea color and texture, green and yellow, smooth and wrinkled. Let's say it got its green and smooth particles from one parent and its yellow and wrinkled particles from another. Green and smooth are dominant over yellow and wrinkled, so the plant will have green smooth peas. Mendel said that both particles for each trait had equal chances to be passed on. This meant that the 4 possible combinations were all equally likely. One crossed with a yellow and wrinkled pea plant, which could only pass on yellow and wrinkled particles, you would get a 1:1:1:1 ratio between each of the 4 possible outcomes. Notice that half have the same trait combinations as their parents, and half have new combinations. These are called parental and recombinant, and will become important later. But what if particles of different traits were on the same chromosome? That would mean that if one of the particles was inherited, the other on the same chromosome would be too. Going back to the pea example, if a green particle was passed on, then a smooth particle would have to go with it. If a yellow particle was passed on, then the wrinkled particle would tag along. This would mean just 2 combinations, specifically the parental combinations from before. While Mendel said that both parental and recombinant offspring would be equally common, the chromosome theory points out that in certain cases there could only be parental combinations. So, the chromosome theory and the Mendelian theory could not coexist. To investigate, Morgan experimented with fruit flies and showed that the particles of inheritance indeed tended towards parental combinations. This tendency for particles to stick together, or linkage, seemed to support the chromosome theory. Mendel, indeed, was wrong. But there was still a problem, since if 2 particles existed on the same chromosome, you would expect no recombination whatsoever, not the reduced recombination that Morgan observed. It was like the recombinant flies had half of one chromosome and half of the other, as if there was some sort of chromosomal crossover and exchange of genetic material. Morgan realized that this sort of crossing over had actually been noted under the microscope, and that this could explain his observations. But this was just a theory. Morgan didn't have any hard evidence for this, all until university student Alfred Sturtevant came along. Sturtevant looked back to the data from Morgan's fruit fly experiments and calculated the frequency of recombination, or how often recombination occurred. Let's call this number the linkage number, since it describes how much linkage 2 traits exhibit. He found that he always found the same linkage number between the same 2 traits, but different linkage numbers when comparing different pairs of traits. Using this observation, together with the chromosome theory, he developed the following hypothesis. For 2 traits to be recombined, a chromosomal crossover must occur in between the particles of the trait. If the particles were close together, there'd be less space for a successful crossover, making recombination less likely. Different traits with different particles had different positions on the chromosome, and so different linkage numbers. The lower the linkage number, the closer the particles. Sturtevant decided to use the linkage numbers to map out the chromosome. Here's how he did it. If body color and wing type show linkage of 17%, and eye color and wing type show 8%, Sturtevant predicted that body color and wing type must either show linkage of 9% or 25%. Exactly as his hypothesis predicted, Morgan's data showed linkage of 9%. Eventually, he was able to make a map of a fruit fly chromosome, showing the precise locations of particles of inheritance, well before the discovery of DNA. Genetics showed that these particles weren't just theoretical, but physical vehicles of inheritance, riding on chromosomes and passing on traits from one generation to the other. They weren't just particles, they were genes. But what are these genes made of, and how do they contribute to the formation of traits? That's where biochemistry comes in. The discovery of DNA as the stuff that genes were made of revolutionized biology, giving new meaning to the ideas of Mendel from a century ago. With it came many important discoveries, all of which point towards a gene being not just a vehicle, but a blueprint. What we found was that in every cell of your body, your genes are highly active. In the space of seconds, the DNA double helix of a gene is unzipped, creating a template for a complementary strand to form. This RNA strand is like a negative copy of the original gene, and is interpreted by a molecular machine called the ribosome into a string of amino acids called a polypeptide, which then folds into what we call a protein. Small differences in how proteins are coded for can lead to large-scale effects on the organism. For example, changes in the OCA2 gene can result in a protein that is less able to play its role in melanin production, leading to less melanin in the iris, and so a lighter eye color. This shows that genes don't just stop after being passed on to your offspring, but continue to oversee their biological activity. They are both the vehicle of genetic information and the blueprint for cellular function. But what I find even more interesting is how genes could be the origin of life on Earth. But first, we need to take a detour from genes and ask an important question. What is life? Here are some features that many biologists agree life must have, but we'll set these aside for a moment and consider the most basic forms of life, all the way at the microscopic scale. Bacteria are alive by most biological standards, but when it comes to viruses and prions, the question becomes more difficult. You've probably come across viruses, nanoscopic parasites that hijack other cells' biological machinery to replicate. Even more basic, but no less deadly, are the prions. All they are deformed proteins, armed with the ability to spread their deformity to surrounding proteins and making them prions too. Biologists usually dismiss both of these pathogens as non-living, because viruses depend on their host cell to replicate, and prions don't have any genetic material. However, there's something deeper that connects all of life with these disease agents: reproduction. Viruses and prions do this in quite an unorthodox way, without using many of the processes and resources that higher organisms use. Still, even at the nanoscale, life's fundamental purpose seems to be reproduction. This leads to an interesting idea about how life first evolved. Imagine an Earth without life, nothing but a soup of chemicals, combining and breaking apart in a random fashion. One day, a special type of chemical arose that could act as a template to form copies of itself, attracting surrounding chemicals to form a perfect clone. Such a chemical, which we can call a replicator, would spread exponentially in the chemical soup, since with each replication, the population doubles. If you think this all looks familiar, that's because this is almost exactly what happens with DNA and RNA from before. Genes may have been the first forms of life on Earth, the very first replicators. But replication involves an energy barrier, a barrier that must be lowered by a catalyst for it to proceed. But how could these basic replicators catalyze reactions? We know that's the job of complex proteins called enzymes today, but could replicators do it on their own? The answer seems to be yes. Folded RNA molecules called ribosomes can function as biochemical catalysts, supporting the theory that genes made of RNA constituted the very first life on Earth. Natural selection would have favored the most stable replicators, the ones most resistant to external dangers. First, they evolved chemical means to create membranes and enzymes and generate energy, but eventually, some of them developed legs, brains, and tongues. This is the RNA world hypothesis, combined with Richard Dawkins' selfish gene theory. There are other theories, but for me, these are some of the more convincing ones. Put like this, life on Earth doesn't seem so special, just a natural product of chemistry. Genes were the first step towards biology, and have been ruling all of life ever since. But there's more to biology than just genes. So to learn more about how proteins and even mitochondria play a role in the complex story of life on Earth, support this channel by subscribing to GeneSpeak and liking the video. This was a GeneSpeak deep dive, and thanks for watching.